Hey there, students. Welcome back. I want to go through the notes that we've had the last 2 days here in week 18. There's a lot that's on the board over the last 2 days, but honestly, a lot of it should be a recap from some of the things we've done previously, so let's jump right in. Up on the board first right here is a picture from the website, which shows the key terms that are up there at the beginning of every week. And remember, you always have some time at the beginning of the week to work on the key terms in class. One of the ones I wanted to point out here, the reason I had it up, is CRISPR. This is one of those things where I'm not quite so concerned that we remember exactly what the acronym stands for, whereas in DNA we do want to know that it means deoxyribonucleic acid. CRISPR, it's more about the technology. In other words, the gene replacement, the gene editing technology that CRISPR represents. And to that end, we're going to go through today's notes and talk a little bit about genetic engineering, artificial selection, and natural selection. So that was yesterday's key terms. Today's notes up on the board. Remember, this is the part of the year where we really get into, I can move my board, we really get into Darwin, the mechanics of evolution. We've talked a lot so far about molecules, atoms, everything that's led up to DNA, and now we're going to talk about, for the next month, the mechanics of evolution. How does evolution by natural and artificial selection actually work? So, we started off with traits being carried by genes. Now in your book, it makes it look like every single trait is carried by just one specific gene. In actuality, oftentimes a trait, hair color, hair texture, for example, when you look at a person, we're going to look around the class tomorrow, for example, at ears, but when we look around the classroom and you see wavy hair, straight hair, something that's kind of in between the hair colors, that's carried by several different genes and many different parts of DNA. So I just wanted to point out that whereas it might show one little part of DNA being responsible for a trait, in actuality, yeah, there are some traits that are carried by just one gene. Those are the ones we'll be checking out in class. Those are the easy ones to codify, and we can do some experiments with them in class regarding, for example, if you have attached or unattached earlobes, if you have a widow's peak, things like that. But it should be learned, and again, I just want to point out going forward that usually genes have multiple places on different pieces of DNA. So that's important to remember. When we look at genetic engineering, remember this is taking a gene from one species perhaps and transferring it to another species. It can also be done within a species. But the example that we're giving in class here is about the glow-in-the-dark cats. They're not truly glow-in-the-dark cats. Remember, it wouldn't be exactly advantageous to be a glow-in-the-dark cat, for one thing. The mice at night would see it coming a mile away. But when scientists wanted to check out how certain traits are transferred from parent to offspring, they have taken the genes from sea jelly. The sea jelly lives a big part of its life in places where little sunlight to no sunlight reaches down deep in the ocean. So you've got a gene that enables the sea jelly to actually glow in the dark, to bioluminescence it's called, and they would transfer that to a domestic house cat. Now, in the real world, you would never have a house cat somehow come in contact with a sea jelly and get a sea jelly gene in their genome. But through technologies like CRISPR, like genetic engineering, they can take a sea jelly gene that causes it to glow, put it into a domestic house cat, and then by looking at the cats, by the way, when they are checking this out, it's not like the cats are walking around glowing. They take one of those artificial UV light, kind of like the black lights that you would see at Halloween time that makes things that are white really, really glow. And you take one of those flashlights and shine it towards the cat, and you can see the gene being expressed. You can see the glowing of the cat. And it doesn't, again, just walk around at night, glow in the dark, although that would be kind of cool. But no, it's not the way it actually works. But when you're talking about genetic engineering, you're talking about mixing genes from one species perhaps to another species, or sometimes between the same species, that you would likely never see in nature happen. We're also going to do a quick recap. Okay, the boards are not moving easily today. But if you check out the quick recap on natural selection, those were the examples that we gave in class a little while ago about the made-up creature, the fuzzy wuzzes. They've got some genetic variety, Their genetic variety has them go from very, very furry to not quite so furry at all. Again, we use them, one, because they're really easy to draw, but two, when you're talking about genetic variety and natural selection, if, for example, the climate changes, say it gets colder in this particular case, the ones that are furrier will have a better chance of survival. Does that mean definitely they're going to survive? No, remember it's odds, it's chance. We're talking about the probability is that the furrier ones that will survive the harsh conditions will be better adapted, will be selected by nature to be able to pass on their genes, whereas the ones that weren't quite so furry might not survive the harsh winters because the climate again was growing colder in our example, and when the climate's growing colder, they might not be surviving the harsh winters. Their genes would be selected against. In other words, these right here would likely be able to pass on their genes. When you're looking at artificial selection, we had two examples in class. One was from a couple of days ago. The great Carl Sagan, and I had linked it down below, so that's from a few videos before, about 2 or 3 videos ago. Check out the Carl Sagan segment if you haven't seen it already on the Heike crab, and you can also find it in several other places online. But these were crabs that their carapace, the back markings on the crab, resembled a samurai warrior, and this was a great example of accidental artificial selection. In other words, the fishermen who would toss these back because they weren't quite so anxious to eat a crab that had a carapace, a back that looked a little bit like a samurai, they would throw them back. They selected for the crabs that had a back that looked a little bit like a person. They were able to pass on their genes, and the genes control the look of the back of the crab. So it was artificial selection in action. Remember, when it's artificial selection, humans are involved in selecting the animals. And then finally, we just had, for example, Thanksgiving here, and when you're looking at farm animals, classic example. Later on, when we watch a little bit of The Biggest Little Farm, we'll see some more of that. But by selecting the largest farm animals, for example, in this case, my poorly drawn chickens, you've got some animals that are selected. When you take the largest ones and you breed them together, you have a better chance. It's not guaranteed, but you have a better chance of getting the larger and larger animals, and you end up with things that you would never see in nature. Later on, when we talk a little bit about dogs, we're going to see how, for example, you would probably likely never see wild packs of chihuahuas in nature without artificial selection. Artificial selection has wildly changed the shape of the domestic dog. And we're going to look at that when we look at, again, go into more detail on artificial versus natural selection. One of the things that the students were asked to do today was a little bit on meiosis. On this particular one, we're not quite so concerned about the details. I wanted to make sure that the students knew simply the steps of meiosis, because tomorrow we're going to talk, tomorrow or Thursday, we're going to talk a little bit about crossing over. Crossing over is the part in prophase 1 where we have the genes dividing in such a way that it lends yet another level of genetic diversity to the genome. So look for that. All right, make sure you get the notes out there online. And yesterday was posted the answers on the website to some of the work that we had in our textbooks. So check that out. Have yourself a wonderful Tuesday, and I'll see you online. Bye-bye.